Hello, good morning, and welcome to the first seminar of the this spring series 2022, the International Plant Breeding Seminar organized here at NC State by the Plant Breeding Consortium. We have the honor of having Dr. Chiedozie Egesi as a speaker today. Chiedozie and I met probably 6 years ago, and I know of most of his trajectory in the last in those 6 years. And he's a senior scientist at IITA, the International Institute of Tropical Agriculture in Nigeria, and he's the director of the Next Gen Cassava Global Project that is aiming at modernizing plant breeding in cassava. Okay. Before that, he was a scientist at the National Root Crops Research Institute in Umudiki, Nigeria, and he has participated in the development and release of 40 improved cassava varieties in Nigeria and in Africa in general. He got his PhD from the University of Ibadan. He has also an adjunct professorship at Cornell in plant breeding and genetics. He has mentored several PhD level students, and he is the president of the Nigerian Plant Breeders Association and a member of the executive board of the African Plant Breeders Association. So Chiedozie, thank you so much for accepting the challenge of opening this series, and look forward to your presentation. I'm gonna stop sharing here. Just a reminder for the audience, as always, place your questions in the chat and through the presentation, and we will address them at the end.
Thank you. Thank you, Carlos. And thank you to your team and to everyone for joining us today. As Carlos has rightly said, I'm talking about cassava. Cassava is an important crop in Africa and even globally. I hope my slide is in the right mode.
Yes, it is. It is in the right mode. Thank you.
Okay, thank you. So we're going to run quickly as much as I can. And you've already mentioned the vision of the project. We are aiming at empowering farmers through innovative, sustainable cassava breeding. If you look to the right, you will see where my cursor is. You will see the different organizations and our partners. There were 13 institutional partners spread across 4 continents. And we, apart from those 13 institutional partners, we have an additional 7 African country national programs that we have formed a growing community of practice, and I'll talk about that. But this project is coordinated from Cornell University, where, like Carlos mentioned, I serve as an adjunct in the plant breeding department and also in the global development department. But the work I'm going to present today is by no means the work of one man. It's the work of everyone that is in the screen in the 4 continents that I mentioned. And you see this is not even exhaustive. But back to cassava, is the ultimate climate change crop. While other crops are really suffering from the effects of climate change, cassava seems to be performing better under the situations that give rise to climate change, elevated carbon dioxide, and increased temperature. And this is beside the crop, the crop being a naturally drought-tolerant crop. So it's very important for today's agriculture. So it's consumed, it's very important for many people, at least 500 million people depend on the crop, and Africa produces more than half of that. Because it's a hardy crop, it's a fallback crop, and you can store some part of it in the ground. But it's also important to know that it's mostly grown. In some African countries, it's regarded as a female crop, while yam and other crops are considered a male crop. So cassava is mostly grown by women and processed by women for home consumption, and it has the potential for increasing the income of those people. But there's been a slow rate of genetic gain in the field. Most of the gain we've had in the past 50 years have been due to increased acreage cultivation rather than genetic gain. And you can see from these charts that we can do more. There's potential to do more, but it's not being realized in the farmers' field. So even though we have varieties that perform better than what you are seeing, but is not really the case sometimes in the farmers field, either because they are not growing them or they don't have access. So what do we need to address that? We need to go back to the breeder's equation, and the breeder's equation are comprised of about 4 components: selection accuracy, selection intensity, and genetic diversity, and the time. So these are the 4 things, and you see the arrows show the areas that we are trying to use to bring intervention. Or maybe to make it clearer, this is what we need to do to make for a modernized plant breeding program. So for the shortening, the denominator, which is a time, we working on 2 things, using genomic predictions and using flower inducing approaches. For selection accuracy, we have a digital ecosystem, and we have managed to work around designs and the analytical pathway. And selection intensity, high throughput phenotyping is very important for us, and we need to maintain genetic diversity to a manageable size that we can manage. But before NextGen came on board, a lot of genomic resources were developed at the global cassava community. In the 90s, we had a few hundreds, and a few hundreds were added a little more in the mid-2000s. But this is pre-NextGen era. But as soon as NextGen started in 2012, we started changing the numbers, and we got to 22,000 by 2015. But between 2015 and 2020 actually, or 2021, we were able to ramp this up to 20 million SNPs derived from over 1,200 cassava clones. And this is important for us. And we have a pipeline now that we've been able to do regular genotyping across the globe of over 40,000. And still talking about genomic resources, we have a way of going from leaf tissue from breeding programs, using Intertech, a service provider. So it tells you that within 8 weeks, we're able to develop our breeding values, genomic estimated breeding values, through the system we have been able to work out right now. I'll keep moving. But cassava breeding cycle typically takes between about 7 years to complete when you are working through the traditional or the conventional method, and you move from the crosses seedlings, and you do a couple of years evaluation before you're able to determine the parents that are good. And this will be as a result of this lady is making crosses, but those crosses will produce very few seeds per cross, maybe 2 per cross. And again, the planting material is a stem, very low, 1 to 5, compared to maize that's 1 to 300, and also an intensive phenotyping. But with genomic selection, we've been able to cut down this, and we're able to cut out this area in red that you saw in the last chart. We're able to determine the breeding value of the parents within 1 or 2 years, and we've been able to demonstrate this very well through the project, through this project, and that's very important for us. Remember to reduce the time to the next to the next cycle is important for us to tweak around the breeder's equation I showed you earlier. So with that, we've been able to rapidly move since the past 6-8 years, move some cycles now. These are cycles which are like completing the years for different breeding programs that are involved with us, including in South America and Africa. Now, I also said to move around the time, you need to work on the flowering. Many important cassava genotypes flower do not flower at natural conditions. And it's been impossible to tap the genes that come from there. So we worked on photoperiod manipulation, plant growth regulators, and pruning, and we've been able to work to see that we can produce in combination, we can have 15 times more, many more fruits and seeds produced. Look at this. This is not very usual, especially in cases where you have limited or shy flowering, as we call them. So we're able to work, study, have understanding of the factors that govern flowering and be able to manipulate them to increase flowering. I also mentioned that to increase accuracy, we need to, to improve on the breeder's equation, you need to improve accuracy. And one of the things we have started using is cassava base. I believe the sweet potato group in NC State is also having sweet potato base, which is very similar or very close to this cassava base. Is a digital ecosystem used for managing cassava, cassava work, breeding work, cassava improvement, from phenotyping, genotyping, field layouts and designs, analysis of the data and selections, and the decision making is all in one ecosystem that is digitized. I would like to say that our cassava base is fully open access, and anyone can access any data that you need from there. A few more information about cassava base. Right now, we've grown it to over 1,300 users from 24 programs across the world. 500,000 accessions are already, more than 500,000 accessions are already there. And all this is important to see that we have grown it to 18 million phenotypes now, and is still growing. So this is a progression from 2018 to date, when we had like less than 10 million phenotypes to 18 million within 4 years. And we're still working to improve on this database and to populate it. But there are things around quality that is important for database management, pedigree information, and you see we've been able to improve on the pedigree information available for the different accessions that are in the different breeding programs. And we have this pedigree string that will tell you, if you click on this genotype, you'll be able to see where it is. And we've been able to also analyze what's the progress. So we see people, no, this is not correct. This is better than this. I didn't update this, sorry. So, for Tanzania TARI, but for the other ones, they are correct. So you see the numbers are improving as we are going. And this was something people were not paying good attention to. And you need the pedigree information for you to say, I have a modern plant breeding program. One of the other things we've done is to have a better understanding of the trait genetics. So if you go to anywhere, Google Scholar or ResearchGate or anywhere, the academia also, and you Google or you search genome-wide association and it is cassava, maybe 90 to 95% of the work that comes from there are all next-gen supported work. And so that's what we've been able to do to make sure we understand clearly whether it's resilience traits, nutrition traits, quality traits, or otherwise. And that we have been working on. And I give an example here where we've been able to determine the genetic signals, controlling cyanogenic component of the roots and that of beta carotene. And this is important, especially as we're running a biofortification program on the side. We'll be able to say that you don't need to process a biofortified crop extensively, but you also notice that the farmers, the local people in Africa, have depended on extensive processing to remove the cyanogenic potential of the roots. So we need to now manage it by having, using the genetic signals to be able to remove the cyanogenic potential of the roots, as an example. Again, I'll move to resilience breeding for virus resistance. So in Africa, there are basically two major viruses that affect cassava, the top photograph and the bottom one. The top one is called cassava brown streak disease, caused by cassava brown streak virus. And the second one is the cassava mosaic disease. You see these mosaic patterns on the leaves. These two are deadly. And when you have them in combination, the farmer is not just on 0%, it could be -% value that they will get because they would have spent money to produce nothing. So it's very important for us. And we are, this disease on top is only in East Africa, Southern Africa, and Central Africa, not yet in West Africa. So, but we are not waiting. So the project is doing preemptive breeding for this in West Africa, while we're also doing the breeding in East Africa. We're also developing high throughput screening methods, and we're working with advanced labs like Stefan Winter's lab is DSMZ in Braunschweig, Germany, and we use that as an avenue for intercontinental germplasm exchange. And the work we've been able to do tagging genes for CMD resistance have been beneficial to Southeast Asia, even though they are not primarily members of the project, but we've been able to share our tools for them to work against cassava mosaic disease in Southeast Asia, which is is less than 10 years it emerged there as a big problem. If something is happening here to my screen. Sorry. So let's talk more about NextGen, and it will lead us to the next couple of things I'm going to be saying. So NextGen is not organized like a regular project where you have work package 1, 2, 3, or 4, or objective 1, 2, 3, or 4. We have divisions that try to mimic what a seed company will do. So what, having a cassava breeding Inc. as in mind when you're looking at this cartoon. So we have the breeding division, which is the center of everything, developing populations and varieties. But we have the research division that also we need to develop new tools, new traits, new protocols. But more importantly, we need to do the survey division of the demand-led component where the needs of the users, the needs, preferences of the farmers or processors is important. And working together, we're able to generate varieties that are really going to be adopted and that can, increase genetic gain in the farmer's field if this is done efficiently. And this is what we've been trying to do. The survey division helps us to determine product profiles that feeds into the breeding goals. So what I just said is to say that we are customer centric in all that we do. I'll keep running because I said time. Okay, so when you look at the work we do, I mentioned that we work with the end users, but we use citizen science to make that happen so that we'll be able to identify the key traits, and they also help us to identify the winning varieties in their farm. So in Africa, but the information here is specifically for Nigeria, but the same similar thing is happening at the same time in Tanzania and in Uganda. So in those countries, we have, for example, in Nigeria, we have 320 farmers for a tricot model. Tricot is triadic comparison of technologies. So each farmer evaluates 3 varieties, as you can see, for variety A in this woman's farm, variety B, and variety C. So she's going to tell us from this harvest, she's going to be able to tell us from her evaluation which one is the best performer and which one is the worst based on what she's looking at. And we'll take records and then we're able to make analysis. And in selecting the farmers, we are very cautious to make sure that there's a social diversity, including gender, is part of our criteria for selection. And the results will come in the next couple of slides. But let's move to this work that we are doing. I mentioned Stefan Winter's lab, DSMZ. We're able to exchange germplasm or breeding materials across the continents, the 4 continents, actually. We're able to, by having a transit port in Germany, where he's able to do high throughput screening for the viruses and the other diseases that are of importance, so that we don't complicate matters or we don't cause more problems while trying to solve another one. We also focusing a lot on the root quality traits. Again, this will resonate with the people in the Craig Yencho's lab in NC State, where they work on sweet potato. Roots quality is very important. Culinary qualities, eating qualities quality is important to us as much as it is to the farmer or to our client. So we've paid more emphasis having better tools to analyze that and to be gender responsive, making sure that the voice of each gender is included in what we do. And again, this is a scorecard for NextGen, showing the genetic trends that we've had over the past 5 decades, from 1970, that's till 2010. We were able to look at the yield, the dry matter, and the disease resistance, how much progress we are making in the last 50 years compared to the time of NextGen. So this is from like 2012, how we've moved, and we've changed the curve right now. And we hope that it keeps going up. Even if it's getting flat, we know what to do by walking around the training population for our genomic selection. So we've been able to increase the rate of genetic gain for cassava. But as to arrive at that, we started by segregating our stages of the breeding flow into different stage gates, starting from product design, which I mentioned is like engaging the farmer, the end user, to determine what you need to breed for, and getting up to official release of varieties. So for some of our programs in Nigeria and in Uganda, we've been able to complete this cycle. And one important thing we've been able to do is to make sure that each year we are having annual product advancement meetings. This is not used to happen in the past, but we've been able to make that happen in public breeding programs. All the cassava breeding programs, all the major cassava breeding programs in Africa are now having annual product advancement meetings. And that is where decisions are made as to what clones or what varieties are moved from one stage to the other. As we move from 20,000 seedlings to arrive at 5 for release, what are the guiding criteria or decision-making tools that help you to arrive and to discard one and keep the other one? So to have an advancement meeting, we will have data, we will have every program aggregate or arrange their data in such a manner, and we'll be able to determine. The more green we have here, the better, the more desirable it is. The more red it is for that trait, the less desirable it is. So you're able to classify or categorize the varieties according to that. And you also do some other biplot analysis where you make selections based on what you are looking for. And these are the criteria or the basis upon which you can recommend that we advance this variety to the next stage. So that is already being done. The other thing we've done in NextGen is to look at the process management system. How do we flow? So it's not we thought it was easy, but we needed to arrive at certain things. So we needed to assemble how to operationalize the process for advancement and how to get there for a breeding program. So we needed to have crop and advancement calendars, standardized roles for each person, because if you're coming to that meeting, the advancement meeting, we need to know what you are bringing on, or I mean, what is your power to make a decision? Are you a critical person in the decision making, or you're just there to be informed? So everybody comes with a determined role, and that's very important to us. So we've been able to have advancement calendars. We've been able to map out the roles of each person in each organization and the decision rights. That's if you're able to recommend or agree or perform or just give input. Like I mentioned, for Africa, we have the cassava advancement calendar now. So in this calendar, we've been able to determine when it is appropriate for you to have an advancement meeting based on your crop calendar, based on how the farming practice in your region is. So we have for Central Africa, East Africa, West Africa, Southern Africa, and some other areas like even though Tanzania is East Africa, but there is a lake and highland area that is different from the general area of East Africa. So we'll be also able to map the key people that are involved in the NAS or in the CGs, in ITA, with unique roles, disciplines, and the places where they fall into. So you can, when you look critically, you will see that each organization has its own, the NAS has its own, the NAS is a combination of all the breeding programs in Africa specifically. We will also be able to do a cassava pipeline process map to the CG NAS stage plan, where we say it starts from stage 0 to all the way stage 7 with all the teams indicated, whether you need a limited team or all the teams, that's or just full cross-functional teams. CF is cross-functional. So we've been able to make this so that clearly we will know where to have an advancement meeting and where to make critical decisions where you don't need everybody. So you have a limited cross-functional team, maybe just the breeders, a few breeders, or you need both social scientists, food scientists, and breeders. So that we've been able to map, and everybody has come to agreement to work with this. And these are other critical steps we need to take for a stage-gated advancement process. Like I mentioned, we've been able to also map in a more detailed version. You will see actual names mapped to all these, whether they're for just information or for action to be taken, as it were. So in 2 years ago, we were able to release 5 varieties in Nigeria, and this will be the first time varieties are released in Nigeria or in Africa according to product profile classes, whether it's for fresh root use or gari and fufu, the most commonly used one in Nigeria or in West Africa, or for industrial, like for starch. So we've been able to do this in Nigeria. In Uganda, we also have, I think in the next 5 months, we'll be ready to meet with the Variety Release Committee of Uganda with these 3 varieties, Narocas 3, 4, and 5. And we have also determined their product profile and that they are serving. These are all serving Nkumba, UG 12156 and 193, all from Uganda, important for the rest of East Africa also. So I mentioned earlier on that cassava has a low multiplication ratio. So one of the bottlenecks when you release the varieties is how do you ramp it up in terms of technology launch and delivery to farmer. It's been a big issue, but we've been able to solve it with our partners in the BASICS project. Is a seed system project, and we've been able to also have seed companies that are clearly solely focused as a business strategy on cassava in Nigeria and also in East Africa. And we have other projects that are supporting to make that happen. So we have a semi-autotrophic hydroponic system that we use to rapidly multiply cassava without going through hardening. You don't do tissue culture, and you can take them directly to the field. This is one big thing that we've been able to use to rapidly scale up new varieties. The other thing that we are really proud of talking about in NextGen is our capacity building work. Students, Mercy is one of our grad students, was one of our grad students who is now responsible for ITA's cassava seed system. ITA has a seed company, and she's really manning, or she's in charge of the operations. She's Nigerian. Ismail is from Uganda, but is one of the strongest postdoc in cassava breeding that you can think of in Africa. So he's been very supportive to the cassava breeding unit in ITA. Lydia is trained by the project and is supporting the national program in Nigeria also. So, and we have many more students. We have over nearly 40 grad students that we have, more than half have graduated. I also mentioned before 7 countries that are involved with NextGen, but are not the main partners. Is the spirit of none should be left behind, is a spirit of spoke the hub approach. So it's a community of practice where we have them to share learnings and resources with us, learnings on regarding to analytical and all the other things like the modernization of plant breeding or cassava breeding by having peer-to-peer visits and safe germplasm exchange, but importantly, to make them be able to use cassava-based digital ecosystem to, design their trials, manage their trials, and even manage data. We've been able to achieve this in part in these countries at various levels, and it's still a work in progress. How are we achieving that? We are holding several trainings. I think this switched from face-to-face training from 2017 to 2019, but from 2020, we began to do more virtual. And we hope that we have a few more face-to-face trainings in this very year, 2022. But this is a work that we're doing to make sure that everybody, we socialize cassava base, we socialize our field book, and we socialize quality control system and analysis. I'm going to be talking about quality control shortly. So we've built capacity at the national program level. So they've now focused on high throughput phenotyping, which is something you left for only the CG centers in the past. Now they can also use field books and evaluate root quality trace, do more than just measure yield in the field. I mentioned about quality control. We call them quality champions. Is aimed at increasing accuracy. And what we have done is we have a quality management system that we've been able to develop where you plan, do a check and act, and then plan again. So we've developed and implemented standard operating procedures and all the things that we need. We've trained, people are more conscious now in quality management systems. And what have we been able to achieve? All our phenotype trial layouts are designed on cassava base. All our field book pheno apps is used for data collection only. We've been able to standardize trait names across all breeding programs, cassava breeding programs in the world. We also have a QC SNP panel for fingerprinting. You can do a light or deep with 18 or 36 SNP markers. And all these other, I don't want to list all of them, but we've been able to see improvements in our data quality. I think I've missed one slide, but it's okay. I wanted to show us about how we've improved the evidence that the data, the quality of data, the metrics of data is showing that we've made, that the improvements in QC has worked. Now, the question is, why is NextGen Cassava Breeding Project a successful model? Is a group that is, we're a cohesive group. We started with few people, but we had a big picture in mind. And the big picture is that we should treat every partner as equal. So, and the good thing is that it was even the bigger programs that were pushing for the common good, for every partner, whether big or small, to be treated equally. And we grew together with Excellence in Breeding. Excellence in Breeding is a CG program that is meant to, increase excellence in breeding. And our project started before them, but we've been able to grow together. They use projects in different countries for demonstration. Most of you know this guy here. His name is Carlos Iglesias, and he happens to be a member of our external advisors, and he's also the convener of today's seminar. So we are being advised by one of the best, or a group of the best. Another person is here, Dave Maya is also here, and is formerly of Corteva. So, and they helped us to focus on quality control. They brought that out clearly to us based on their former experiences in these different seed companies they had worked with. And they introduced us to the importance of collaborations and consistent, to keep our eyes on continuous improvement. If you improve, keep improving. And that's what has helped us. So we've been able to work on those quality control measures adopted a demand-led breeding system, and be very open-minded about how to work, and open-minded about areas that we can still improve on. Going forward, as I begin to round off, so people ask, okay, the project is coming to an end by exactly 31st of March 2023. And that's 1 year, 12 months from today. So what do you hope to be doing? So we still have a lot of grounds to be covered. Enviromics is a way of integrating agronomy and genomics and breeding together to bring about crop improvement. So we are looking at that. We're looking at bringing in more high throughput phenotyping tools that are being developed and that we are part of. We want to do better virus screening, even where there is no virus, and to do more allele mining for resistance, transgene, gene edited traits integration into our breeding population. It is something we are very open about, and to continue focusing on population improvements. We already have a HapMap that we are beginning to test, but we want a more robust one that we can have transcriptomics included in it. And that's a work in progress by our team at Cornell at Buckler's lab, and to have a better seed systems and to have genomics that is able to deal with deleterious aspects of the genome of cassava. And we also want to do a little more on our demand-led breeding, where we don't just make it what women want, but what everyone wants. What's shifting from just gender focus to thinking more about social differences. I mean, there's a difference between what a rich woman who has a kitchen wants and that of a poor woman who is in the village and just needs to eat. They probably don't need the same cassava varieties, for example, and other gender classes that are there. Methods to survey, so methods to achieve those, to determine what are the needs and preferences, we need to keep refining, and to unpack the market. What does it mean? And when we're talking about market, like a seed company like Syngenta or Bayer, is different for them compared to when a public sector breeding program is talking about the market. They are not the same because there are many differences. We hope to continue as a flagship breeding program for the 1CG and to represent RTB. RTB means roots, tubers, and bananas. Sweet potato belongs here too. And we want to model the kind of partnership that we've had between advanced labs, CG centers, and national programs. This is not an easy partnership to manage, but we've been able to show that it can work if it is properly managed. And we'll continue to work with those advancing centers and help to design and link to sustainable seed systems capable of the last mile delivery, because it's very important, the last mile delivery. So what are our take-home lessons? With the right tools, even challenging crops like cassava will respond effectively to selection, even within a short time. In an efficient manner, you can breed cassava. We have made great strides in modernizing cassava breeding, and great strides in the technologies that helped us to achieve that, and in our attitude and in our focus. So we changed attitude from moving from focusing on varieties alone to population. We are working on population improvement, moving the population on the path of continuous improvement. And we are focusing more now on the root quality traits, the cooking qualities and all that. We continue to innovate because there are smallholder farmers hopeful for new opportunities from our varieties, which are obviously better than what they been growing. So current signages and partnerships, we want to, like I mentioned, we want to have a better understanding of how agronomy and the environment is important, going to help us select the best, even when we're saying we are doing genomic selection. I mentioned it before, enviromics is a fairly new approach that we hope to develop a platform, a next-gen cassava platform for, and we'll be able to predict unknown environments and from there. the work is new, and we are beginning to think around this for our next phase. We can boast about being an extensive plant cassava breeding network. We have outreach in different continents, like you can see here, but we're the largest breeding cassava network, if you think about that. We also think we're the largest variety testing network for cassava. We have a lot of collection, especially through our CG centers at CIAT in Colombia and IITA in Nigeria, and even our partners in Uganda for cassava with native traits. And we have the largest database for any, whether it's phenotype, molecular, genotype data for cassava. We have capacity for field testing of even gene-edited or GM crops, and we're very willing to partner with you. We have also, so it suffices to say that modernized plant cassava breeding is essential to Africa's food security future. And I want to thank you for joining us and for listening to this. Maybe I have some questions if there are any. Carlos.
Hey, Chidosi, thank you so much. Very good presentation and seminar. And it's nice to see a lot of familiar people in this seminar, and nice to see an attendance from all over the world. There's already a couple of questions in the chat, but I wanted to stress a couple of things that what you said about open to collaboration. One of the areas that we are working here at NC State is trying to develop gene editing methods that are more genotypic independent and species independent. And we are interested, since we are having some model species as sweet potato, we would like to include cassava in that study. So there's some graduate students and postdocs working on that, and we'll probably reach out to you on how to get some materials that are a good reference to include in those studies. Okay, so that's a specific area. The other thing is that I apologize to you for getting to mention that you were the recipient of the 2021 Achievers in Agriculture Award in Nigeria, which is the most prestigious award in terms of innovation in agriculture in Nigeria, and that's amazing, Chidosi, for a person that we know the trajectory is amazing, and congratulations on that. Okay.
Yep.
So going to the questions that we have in the chat, the first one I see is Claire Hershey is asking, what is the sustainability model for funding of the next gen activities? Will you shift towards internal support from national programs? Or you continue to depend highly on major donors? What is your take?
So our goal is to shift towards a national program focus, but I think we still have a lot to fine tune to show the progress we have made. For example, the demand-led component. So our strategy is that as we engage the local partners, they will begin to see the value in what we do. Right now, it's not just the national program, because some governments don't even care. So we're looking towards a public-private partnership model where the seed companies will be able to take off some load and provide some support to some end. For example, supporting the product advancement meetings, for example, supporting some on-farm trials with their farmers, and joint, like we have demand creation trials that we are doing with NextGen. I didn't mention that, whereby new varieties are tested by seed growers. And some of these seed growers are also linked to industry, as cassava-based industry. So they will begin to not just provide cash, but they're able to say, don't worry, just give us the planting materials, we'll pay for the trial. And that would be a good step. We've started that but we need to deepen it the more. But at this stage, I don't think is the time for the donors to hands off. It's still too early, but is a journey that has begun. Thank you.
Okay, we have several messages praising your presentation and the approach of the project, the Cassava project. Okay, we have a question here. Just right. Okay, he says, "I would like to know more about environomics. It's a new subject. Do you have plans to expanding collaborative efforts to see how the disseminated lessons of Next Gen Cassava project can apply with other crops within this food system?"
Okay, so I would like to start with the lower one, the later part, because it's the easier one. So we've already started collaborative efforts, and we are working with we are working together as RTB as food system, roots, tubers, and bananas food system. But for the R&T, we work particularly with the sweet potato team, including the ones in NC State and the ones in other parts of the world, Peru and East Africa. We're also working with the Africa Yam. So they are, because we have a lot to share in common in terms of phenotyping, in terms of the genetics and the behavior of the crops, because we also have the similar vegetatively propagated seed system model. So we're sharing resources at that level between NextGen and those partners. We have other partners that I may not have mentioned, like working together with the Africa Cassava Whitefly Project, where we are looking at breeding for resistance of viruses indirectly by breeding for resistance to the vector. So all the information or the products that are coming from those partners or projects are feeding into ours. And we hope to improve and scale that up in the coming years. Concerning enviromics, like I mentioned, is a new area where you're not just talking about improving the genetics of the crop. A lot of times some breeders will present the case simply. Once you get the genes for high yield, or genes for resistance, or genes for anything, then you are good to go. But sometimes the environment has an influence. So you may breed something that is very good, but the environment will not let it be expressed. And we need, now that we are talking about genomic selection, genomics assisted breeding, bringing agronomy to play a role and see how the interaction happens is what we have not been doing, or because genomic selection is relatively new. So this is an area that we hope to put some emphasis on as we move forward. Thank you.
Okay, I have another question here from Ernie, is asking if you have identified traits, targets for gene editing in cassava, and if you have identified lines that will be lead candidates for further research in that area.
So is that a question?
Yes. Have you identified trait targets for gene editing in cassava?
There are a few groups across the world that are doing gene editing, but critically, if you ask me, I know the crops that we need, we have issues with. I know the traits that we have issues with that I think we can focus on. One of them is post-harvest physiological deterioration, and that has really not been, we've not made a lot of progress on that. The second one is a disease trait, a bacterial disease that we need resistance to. We have also not made a lot of progress on that, cassava bacterial blight. But a few people have worked on using gene editing models to reduce the cyanogenic potential, and that's important for areas in the world where there's not extensive processing of cassava roots. So it's important for us, and it's also important if we want to increase the use of foliage, the leaves of the crop to form livestock feeds. That's a big win. Also important, like I mentioned, for biofortified varieties of cassava.
Okay. Yadira is asking, do you have specific needs in terms of high throughput phenotyping, and what kind of collaborations are you building around that?
Yeah,
I
like
imagine the underground
exactly.
phenotyping.
Ground penetrating is the first one. It's a work in progress, but I mean, that's where we will need all the help for funding the work and all the help for bringing technology to help power that, whereby you do non-destructive determination of yield for evaluations for the roots that's underground still while the plant is standing. So you'll be able to know the potential yield of the crop before it matures and before you harvest, just by running some probes under the soil where the roots are growing.
Right. Any initiatives with cassava water use efficiency phenotyping? That's a question from Larissa.
Any what?
Initiatives related to cassava water use efficiency.
A few work has been done. Water use efficiency in cassava is not as easily measured like it is in other crops, because while you're looking at the growth, what's most important is the economic part, which is the roots. And so again, it takes you back to yield. So how does it yield compared to when the water regime is normal? And this is different from all the ABA and all the salicylic acid that people measure for other crops, because those do not correlate very well in cassava. But not a lot is being done in that regard right now, and that's an opportunity that exists. I agree.
Steen Hoyer is saying that he likes to hear more about the 55K images in cassava base. He says sounds like a tremendous resource. Are they mostly phone photos?
Yes, they're mostly phone photos. A few are camera, but mostly are phone. Just any technician just goes to the field and takes the photo and uploads it through cassava base right there on the field, and it's uploaded to the web, to the database. And you can access it, Steen. You can access it even as Steen. I mean, right away, all you need to do is to open an account in cassava base, and you can have access to all the photos. And there's an email for interaction. You can also email me. I'll direct you to the relevant person to talk to if you want to do more on that or work with that.
I'm posting your email address if that's all right.
Yeah, that's fine.
There's Chidosi's email address at Cornell. Any other questions before we close the seminar? Here we have another one. Are you planning to apply double haploid technology with genomic selection in cassava?
Femi, I don't know. That's not in my immediate plan, but there are a group in SIAC that has been working on this. I bet we need to first get the double haploid technologies first working before we say apply. That would be the second. So you've jumped to the second stage, but we need to even get double haploids. We don't have it yet for cassava, but I know it's possible. So, but we need partners, we need sustained funding to make that happen also.
It's an important question. Linia is asking, how open is cassava base? Do you have to be part of the cassava community, or can any scientist working around the world access cassava? Anybody. Okay.
Anybody on planet Earth can access cassava base.
Okay. All right. Well,
that's free. Good. Yeah.
David posted a comment and says, I'm glad that you have focused on not just the genetic gain, but realized genetic gains in farmers' fields. Great presentation. To me, having witnessed the evolution in cassava, next-gen cassava, one of the greatest achievements is the part of data quality that you guys have achieved, not only from the field, but also in terms of submission of samples and right labeling of samples for the genetic analysis. I mean, all that comes to, and it's something that even us at the university, sometimes we struggle with having good quality data, and you guys have made strides in the evolution towards high quality data. Okay?
Thank you, Carlos.
It's easy to have central databases and to have quick access to data, but if the data is not good, then it's not worth it. Okay, so you guys have done a great job. Okay, so I don't see any other questions unless Thiago, do you see any other questions in the chat that I have missed? I want to double check because there's so many messages coming in congratulating you, Chidosi, and praising your presentation. Okay.
Thank you.
All right. So let me finish this seminar. Thanking again, Chidosi, for your excellent presentation about NextGen Cassava. It's been a pleasure to have you as a presenter, and also a pleasure to work with you throughout these years. Okay. I just want to remind the audience that we will share the recordings from this seminar between today and tomorrow. A special thanks to the support that I get from Brandon and Thiago for organizing this seminar. And just to announce that next week at the same time, we'll be hearing from Dr. Juan Acosta, who is a professor here at NC State, about somehow a similar approach in a cooperative group doing tree breeding and genetic gene conservation. It started as a project in the Americas, and now it's a global project. It's an amazing, very interesting approach that I hope you guys can join us next week. Other than that, I hope you guys have an excellent week ahead, and thanks everybody for joining, and I look forward to have you again with us next time. Okay, so
thank you.
one thing I think I didn't share the screen for the last part. Let me share here. I think it's shared now, isn't it? So that's,
yes.
That's the announcement for next week. Okay. So other than that, thank you so much, and I hope you guys have a nice week. Thanks, Chidosi. Have a good time.
Thank you. Bye.
Bye-bye.